Assalamualaikum warahmatullahi wabarakatuh. I am Nurhani Sakinah binti Halim, metric number 1170851, and today I will be presenting my proposal title, Synthesis of 4-methyl coumarin derivatives as antioxidant. First of all, introduction. In recent years, there were abundant evidences suggesting that free radicals, especially reactive oxygen species, involved in many diseases such as heart disease, asthma, and even aging. Due to this situation, many researchers devoted their study in order to synthesize novel antioxidants to prevent these diseases. Coumarins are one of the sources of antioxidant. Coumarins are easily found as they are widespread through the plant kingdom. Coumarin can be found in plants such as sweetgrass, tonka beans, strawberry, blueberry, and even apricot. Coumarins are popular due to their biological characteristics such as anti-cancer, anticoagulant, and antiviral. In this study, parchment condensation will be used to synthesize the coumarin. Parchment condensation is the synthesis of coumarin using phenol and beta-keto ester under acidic condition. Free radicals are actually the byproduct of aerobic respiration and they are present around us. Free radical is a type of unstable molecule that contain oxygen that easily react with the cell in our body. The buildup of free radical, especially reactive oxygen species, in our body can cause damage to the cell, DNA, RNA, and protein, and this will further cause into aging process and fatal diseases. Therefore, many studies were conducted to find a simple way and efficient to synthesize antioxidant from coumarin derivatives. Parchment condensation is the most popular method to synthesize coumarin as it is simple, efficient, and economical. Other reactions such as Perkin reaction is a bit complicated as it is difficult to obtain appropriate intermediate product. The objectives of this study are to synthesize 4-methyl coumarin derivatives, to characterize the synthesized compound using microelemental analysis CHNO, Fourier transform infrared FTIR, and proton nuclear magnetic resonance HNMR, and to determine the application of synthesized compound as potential antioxidant. The scope of study is synthesizing 4-methyl coumarin derivatives using parchment condensation, characterizing synthesized products by using CHNO, FTIR, and HNMR, and determining the application of synthesized product as potential antioxidant using 1,1-diphenyl 2-picryl-hydroxyl DPPH radical scavenging activity. Next, literature review. There are many applications of coumarin. A few popular applications of coumarin are as anticoagulant and used in laser dyes. In addition, coumarin have a vanilla scent which make them to be used in perfumes and flavoring in soaps and detergent. To the coumarin derivatives and their biological properties. Coumarin have many biological properties such as antioxidant, anti-HIV, anticoagulant, and antimicrobial. A popular coumarin derivative that have antioxidant property is 7,8-dihydroxy 4-methyl coumarin, or also known as DHMC. 6,3,4-chlorophenyl acryloyl 5-hydroxy 4-methyl coumarin have anti-HIV properties. 7-hydroxycoumarin is known to be anticoagulant, and 3-acetyl 6-bromocoumarin have antimicrobial properties. There are a few ways to synthesize coumarin. The first one is parchment condensation. Parchment condensation is the reaction between phenol and beta-keto ester under acidic condition. It is widely used as it is a simple method and economical. Next is the Perkin reaction. Perkin reaction is the heat-dependent reaction of aromatic aldehyde and acid anhydride with weak base, which is alkali salt. This reaction produces cinematic acid. Next is the Navier-Vinagel condensation. It is a condensation of aldehyde or ketone with active hydrogen compound in presence of basic catalyst. This reaction produces alpha-beta unsaturated compound, but this reaction has its limitation, which it uses dangerous and carcinogenic solvent. And the last one is Wittig reaction. In Wittig reaction, aldehyde or ketone react with triphenyl phosphonium yield, or called as Wittig reagent, and produce alkene and triphenyl oxide. This method is popular to synthesize alkene. Methodology. This is the overview of the methodology, which contains of 3 steps. The first one is the synthesis of 4-methylcoumarin and its derivatives. The second step is characterization of synthesized compound, and the third step is the free radical screening activity of synthesized compounds. The first one is the synthesis of 7-hydroxy-4-methylcoumarin. In this reaction, resorcinol is reacted with ethyl acetoacetate in the span of 30 minutes. After that, concentrated sulfuric acid is added and the mixture is put into ice bath and stirred continuously. The mixture will be kept at room temperature for about 3 hours and then poured into a mixture of ice and water while stirred vigorously. The mixture will be then kept at room temperature for about 3 hours and then poured into a mixture of ice and water while stirred vigorously until precipitation forms. The precipitate will then be filtered, washed, and dried, and it will be recrystallized using ethanol to get pure product. Next, 7-hydroxy-4-methylcoumarin will react with excess bromine to produce 3,8-dibromo-hydroxy-4-methylcoumarin and 3,6-dibromo-7-hydroxy-4-methylcoumarin. In another synthesis of coumarin derivative, 7-hydroxy-4-methylcoumarin will be dissolved in concentrated sulfuric acid and kept in ice bath. When the temperature is below 1 degree Celsius, the mixture will then reacted with concentrated nitric acid and concentrated sulfuric acid. The mixture will then put into ice bath while stirred until crude product obtained. This crude product will be filtered, washed, and dried. This crude product will be filtered and washed with cool water. This crude product will be filtered and washed with cool water for purification purpose. Then the crude product will be added to boiling ethanol in a conical flask. The filtrate will be concentrated and cooled in an ice bath. The mixture then will be filtered over product. The mixture then will be filtered and the product will be 8-nitro-7-hydroxy-4-methylcoumarin, while the residue will be 6-nitro-7-hydroxy-4-methylcoumarin. The 8-nitro-7-hydroxy-4-methylcoumarin will be recrystallized with ethanol and collected. The instrumentation that will be used to characterize the synthesized product are microelemental analysis (CHNO), Fourier transform infrared FTIR, and protonuclear magnetic resonance (HNMR). Next is the DPPH radical scavenging activity to measure the antioxidant potential. For different concentration of the synthesized compound will react with the ethanol of the DPPH. The synthesized compound will then be incubated for 30 minutes. Then, the color of the DPPH is observed. After that, the synthesized product will be run under UV visible to determine the decrease in absorbance value of DPPH. Next, the expected result. These are the expected structure of the synthesized product. FTIR spectroscopy identifies chemical bond in a compound. FTIR work by measure the range of wavelength in the infrared region that are absorbed by the product. Next is the expected chemical shift in HNMR. HNMR is used to determine the position of H and the structure of the molecules. This is the calculated percentage of element of synthesized products, and this data will be compared with the data from CHNO. This is the expected result for potential antioxidant determination using DPPH radical scavenging activity. The color of the DPPH will first be observed. If the color change from purple to yellow, this indicates that the product have the antioxidant capability. After that, the synthesized compound will then run under UV visible to determine the decreasing absorbance value of DPPH. According to previous work, the lower the absorbance of DPPH in the mixture means that it have higher free radical scavenging activity. Is the milestone and project planning. This is the Gantt chart, which the synthesis of formylacoumarin and its derivatives will start in October, sample analysis using DPPH assay in October, and the analysis of result and discussion will be in October and November, and thesis writing will start from October and finish in December. These are the references used in the slide. So that's all from me. Thank you.